Welcome to MLT 101, Human Anatomy. My name is H1 Barnes, and the lesson topic is the composition and flow of blood. Blood has historically been referred to as the river of life, as it flows throughout the circulatory system to deliver nutrients, oxygen, and other substances to cells, and transport waste products away from cells for elimination. Blood is a mixture of fluid and cells, and is about 5 times thicker or more viscous than water. It's slightly alkaline with a pH of about 7.4. It should remain between about 7.35 to 7.45. Depending on the source you check, it may also say 7.34 to 7.44. pH is how acidic or basic something is, with 7 being perfectly neutral, 1 being very acidic, 14 being very basic or alkaline. In the living body, we refer to the fluid portion of the blood as plasma. The cellular portion of the blood is called formed elements. In an average person weighing about 154 pounds, they will have a volume of blood of about 5 liters or 5.3 quarts. About 55% of that blood volume is made up of plasma, and about 45% is made up of the formed elements. When you look at a tube of blood that's been allowed to sit and settle out, so all the cells settle down on the bottom of the tube, it's going to look like it's half cells and half serum or plasma. Most of the time. Normal plasma is a clear, pale yellowish fluid that's about 91% water and about 9% solutes. Solutes is the term for the things that are dissolved or floating in that water, such as dissolved salts or sugars or fats or proteins. The composition of this solute portion includes gases like oxygen, carbon dioxide, and nitrogen, minerals such as sodium, potassium, calcium, and magnesium. The sodium will help maintain your fluid balance and pH. Calcium and potassium balance is necessary for normal heart function. Potassium is an essential nutrient for normal muscle activity and the conduction of nerve impulses. Calcium is needed for proper bone and tooth formation, nerve conduction, and muscle contraction. It is also absolutely critical for the blood clotting process. One of the ways we can have anticoagulants function is to remove the calcium from someone's blood. You do that, you actually stop their blood from being able to clot. Add calcium back in, all of a sudden it can clot again. Waste products of metabolism include things like blood urea nitrogen, referred to as BUN, creatinine, which is a metabolic byproduct of creatine. It is different. Creatine and creatinine are different things. And uric acid. Nutrients that supply energy to the body. Some of those nutrients include carbohydrates like glucose or lipids such as triglycerides and cholesterol. Proteins, of which albumin is the primary protein in your bloodstream. It's manufactured by your liver and functions helping to maintain osmotic pressure. It causes your blood to be concentrated with stuff so that it attracts water. You also have antibodies floating in your blood, which are used to combat infections. There's another protein in your blood called fibrinogen that is also manufactured by the liver. Fibrinogen is that fibrous material that makes a blood clot. It's like a fiber that holds all the cells together and the platelets together as they clot and clump up. Other substances that can be found in the blood are vitamins, hormones, any medication that a person may have taken. The formed elements, the 45%, the red portion of the blood, is mostly composed of erythrocytes, also known as red blood cells or RBCs for short. Most people are just going to say RBCs. They are the most numerous cell in the blood. You've got about 4.5 to 5 million per cubic milliliter. It's a lot. You have a lot of red blood cells. Their main function is carrying oxygen from the lungs to the cells of the body and carrying carbon dioxide back from the cells to the lungs to be exhaled. Your red blood cells are produced in the bone marrow, and as they form, as they mature and grow up, one of the last stages in their growing up process is to eject their nucleus. They're a cell that actually gets rid of its own nucleus, which means once they've done that, they can't divide anymore. They can't create new proteins from the genetic material in the nucleus, they get what they got. And that's about it. Because of this, your red blood cells tend to live about 90 to 120 days, about 3.5 months. They're one of the shortest lived cells in your body. After that, they begin to lose their elasticity. They're not flexible anymore. They start to fall apart. They get removed from the bloodstream by the spleen and by the liver. Because they don't have a nucleus, we call them anuclear. Because they're sort of donut shaped, but like a donut that's got a membrane in the middle, so you can't poke your finger through the hole in the middle of the donut. It's got a thin patch in the middle. We call them biconcave. They're indented on both sides, and they're about 7 to 8 micrometers in diameter. A micrometer is 1,000th of a millimeter. A millimeter is 1,000th of a meter. So they are 7 to 8 millionths of a meter in diameter. Your RBCs have intravascular function, which means that they do their job inside the bloodstream. You also have leukocytes, or white blood cells. We call these WBCs. They do have nuclei. The average adult has about 5,000 to 10,000 WBCs per cubic millimeter of blood. Your WBCs are formed in your bone marrow and lymphatic tissue and in the thymus behind the sternum. They are said to have extravascular, outside the blood vessel function, because they can leave the bloodstream to go fight infections other places in your body. White blood cells may appear in your bloodstream for only 6 to 8 hours, but some of them can, after they leave the blood, live in the tissues of your body for days, months, or even years. Some of them can live up to 10 years. They are one of the longest lived cells in your body. And the lifespan of white blood cells varies dramatically depending on which type of white blood cell you're talking about. The category of white blood cell contains as many varieties in it as if I were to go to the mammal section in a zoo and point at a lion and a dog and an elephant. They're all very different in what they do. But because they look similar under a microscope, we categorize them as white blood cells. Also because they are literally physically whitish in color before they're stained and looked at under a microscope. The main function of white blood cells is to neutralize or destroy pathogens. This can be accomplished by phagocytosis, which is the process in which a pathogen or foreign matter that's found in the body is surrounded, it's engulfed by that white blood cell, and it's broken down inside the white blood cell. There are different types of white blood cells. They can be identified by their size. Some of them are small, some of them are very large. By the shape of their nucleus. Some of them have nice, round, easily defined nuclei. Some of them have twisted and turned and contorted nucleus that looks like it went through the ringer. You can also determine them whether or not there are granules. They almost look like colored grains of sand inside the cell, present in the cytoplasm of the cell. When you stain these cells with a special type of stain called a right stain, some of them will have granules that turn bright orange. Some of them will be dark purple, and some of them will be a clearish color. That can easily help you determine which cell you're looking at. If it has visible granules that you can easily see, we say it is a granulocyte. Cyte meaning cell. White blood cells that do not have visible or easily seen granules, or have extremely fine granules that are just hard to see with the eye, we call agranulocytes. Granulocytes are the ones we distinguish by the color of their granules. We have neutrophils, eosinophils, which are often called eos, and basophils, which are often called basos. Neutrophils are usually the most numerous type of white blood cell in an adult. A typical neutrophil is called polymorphonuclear. That means its nucleus has several lobes or segments. They can also be called a poly, or a PMN, or a seg, which is short for segmented neutrophil. The nucleus of these polymorphonuclear cells is very distinct. You can tell it apart from all the other cells around it very easily. Agranulocytes. There are two types of agranulocytes. We have monocytes, which are called monos, and we have lymphocytes that we call lymphs. Lymphocytes are normally the second most numerous type of white blood cell and the most numerous agranulocyte. The majority of people are going to have more lymphocytes than they do monocytes, but they are going to have more neutrophils than they do lymphocytes. The two main types of lymphocytes are T lymphocytes and B lymphocytes. Something to know about monocytes is they are the largest of the white blood cells. They're more easily distinguished by their size than anything else. Next, we have thrombocytes. They're more commonly called platelets. That's what most people know them by. And they are the smallest of the formed elements in the blood. They are not actually cells themselves. They are a small piece of a larger cell called a megakaryocyte, and you can find those in the blood marrow or the bone marrow. The number of platelets in the blood, called the platelet count, of an average adult is about 150,000 to about 400,000 per cubic millimeter. Platelets are essential in the coagulation process. No platelets, no clotting. Too many platelets, too much clotting. You want to have the right amount of platelets. They are the first cell on the scene when an injury happens. They're the ones that tunica intima has been cut away, platelets respond. They're right there all the time. They're all over the place, ready to go. Because platelets are not an actual cell themselves, they're just a ejected small piece of a larger cell, their lifespan is only about 10 days. Serum is the term that we use for blood that has been removed from the body and made to clot. We remove the fluid, the plasma, off of the top of that clot and separate it away. We keep the serum, that fluid, and that the leftovers in that tube, the blood clot in the bottom of that tube, will be set aside. Serum specifically refers to blood that has been forced to clot and then has been separated into its formed elements and fluid. Serum is the fluid that we pull off the top of that clotted blood. Because it has clotted, the clotting elements and proteins have been removed from serum. We can hold on to serum longer than we can plasma without having to worry about it clotting, without being put into an anticoagulant. Normal fasting serum is a clear, pale yellow fluid. It looks exactly the same as plasma because it is almost exactly the same as plasma. It is plasma that has had the fibrinogen and clotting factors removed. That is the only difference between your serum and your plasma. Blood in the body is made of formed elements and plasma. We take blood out of the body. If we put it in a tube that makes it clot, it gives us serum. If we put it in a tube that has an anticoagulant and prevents it from clotting, when we spin it down, we're going to have plasma because the clotting factors are still in it. That's what distinguishes plasma from serum, is the presence of clotting factors. There are many lab tests that are run specifically using serum just to avoid the potential for that blood clotting or those clotting factors interfering with the internal workings of the analyzer that you're putting that blood on. Because not all tests can be ran using serum, the obvious example being a coagulation test, we do often need a plasma sample instead. Fibrinogen and many coagulation factors get used up in the process of clot formation that makes that serum, and some tests such as potassium and ammonia cannot be performed on serum because the clotting process releases potassium and ammonia from the cells in the blood, which causes the amount that we would detect in our test to be falsely elevated. Because when we look for that, we only want to know how much is freely floating in the fluid part of the blood. If we cause the cells to release those chemicals and then test it, we are getting extra on top of what we were looking for. It's falsely elevated. It's not the real value because it's not just the level of the fluid that we were testing. There are also tests where results are needed immediately. Waiting for blood to clot, even with the coagulating additive in the tube, can take up to 30 minutes or more. And that's just not acceptable. If you've got a patient who's having a heart attack and you need data now to help the doctors, you can't wait 30 minutes for a blood tube to clot. That would be when it would be preferable to run your test on a plasma sample, because instead of waiting for the tube to clot before you separate the blood components, you can separate them immediately. Normal fasting plasma is visually indistinguishable from serum. You will not be able to look at the two and tell them apart. A large number of tests, especially as we go forward in time, can be run on either serum or plasma. Both are equally acceptable for a majority of tests that are ran in a standard clinical lab. Because of this, it is very common to get plasma tubes all day long instead of serum tubes, because the process to collect the samples is identical. The only difference is what tube the blood is collected into, and it will get the doctor a result faster. A whole blood specimen is in the same form that it was in the bloodstream. This means the blood must not be allowed to clot, and so an anticoagulant has to be used. If the specimen is left standing undisturbed for a long period of time, usually it takes about 30 minutes to be noticeable. After about 4 hours, the process is fairly complete. The elements of blood will begin to separate out. The plasma will move to the top of the blood. The red blood cells will move to the bottom, and a middle layer called the buffy coat, consisting of the white blood cells and the platelets, will sit in the middle, in between your plasma and your red blood cells. It's easy to understand why the red blood cells will sink to the bottom, because they're filled with metallic iron that attaches to the oxygen. That's actually what carries oxygen through your blood, is the iron in a protein called hemoglobin. So if you have cells that are filled with metal, they're definitely going to sink to the bottom underneath the other ones. If you allow this to occur, you cannot just test the tube like that. You have to mix it for a minimum of at least 2 minutes before you perform a test if you need to perform a test on that tube. You can do this by hand through gently inverting the tube. Never shake a tube of blood. The act of shaking a tube, especially on blood that's no longer in the body, can actually break the cells, and it'll ruin what you're trying to do. It's better to gently invert the tube. You just tilt it upside down repeatedly for 2 minutes straight, but that's time consuming and tedious, and nobody really wants to do that. We have a machine called a rocker, which will do that for you. You put the tube on the rocker, you walk away, you take care of your other business, you come back 2 to 5 minutes later, and you can run your test.